This video is about an article in the New York Times called Fast Reproducing Microbes Provide a Window on Natural Selection. It's by Carl Zimmer and was published June 26, 2007. The article starts with an innovative new technique to chronicle evolution in action by subjecting E. coli to a stressful environment to produce new traits. The experiment was conducted over 20 years and continues to this day. The article then briefly examines several other experiments that continue in the same method of E. coli cultivation to produce new traits and show evolution in action. Several basic steps of evolution will be explained so that everyone can understand this article and will find it as interesting as I did. I will also go into the writing style of the author and how he organizes the article to make these experiments understandable to people and an enjoy to read. Evolution is the change in genetic inherited traits of a population of organisms. After a population splits into several small groups, these groups evolve independently and may eventually diversify into new species. Ultimately, all life is descended from a common ancestry through a long series of these specific events. All life can be traced back to its roots about 4 billion years here on Earth. The main mechanisms of evolution are genetic drift and natural selection. Genetic drift gives the genome random mutations, and natural selection is what selects these random mutations to be passed on to the next generations. And an illustration of how natural selection can be seen here in this picture. A organism starts at the top as a gray-looking circle. It has genetic drift, which gives it several different mutations in the next generation. Only the darker circles will survive, and the lighter ones will not pass on their genes. As generations go on, produced animals will become darker and darker. Genetic drift will be explained further later on. Here are excerpts from an interview with Dr. Steven Novella, an American clinical neurologist, assistant professor, and director of general neurology at Yale University School of Medicine. Novella is best known for his involvement in the skeptical movement and for producing a weekly talk show podcast. Dr. Novella will be talking in more detail about the first E. coli experiment talked about in the New York Times article.
Evolutionary biologist Richard Lenski of Michigan State University has been growing E. coli in his lab for the last 20 years. This is a pretty long-term experiment he's got going on there. He's been growing 12 different colonies of E. coli through 44,000 generations, testing some theories of how evolution unfolds over time. What he specifically did was he put them into a very low glucose medium so that they could survive, but they couldn't thrive. They were under stress from marginal amount of food that they were being given. But he did put a lot of citrate, which is another potential source of carbon that some bacteria can live off of, into the medium. But E. coli can't eat citrate. They don't know how to get it into their cells. So here we are. The E. coli are able to survive just barely, but they're bathed in food that they can't eat. And the question was, how long would it take, or could they evolve the ability to get and metabolize the citrate? And then they wanted to find out just what evolutionary process would that be, and specifically, how would the 12 lines be compared to each other?
Dr. Novella, how do they compare each of the 12 lines with each other?
So what he did is every 500 generations of each of the 12 lines, he froze the bacteria and preserved them so that you can go back and then and regrow the bacteria from any point along that line. So you literally could rerun the clock of evolution in these 12 lines.
Can you explain the results of this experiment, how they're examples of evolution, and why it's important?
Well, this is what happened. One and only one of the 12 lines eventually evolved the ability to eat citrate, and they began to thrive and grow and diversify. The other 11 didn't. It turns out that 3 separate mutations were required for that new ability to arise in the E. coli. There was one mutation which did not apparently provide any new ability or any advantage to the E. coli. It was just genetic drift. It was a neutral mutation, probably. But it paved the way for a subsequent mutation that enabled the bacteria to eat the citrate a little bit. And then there was a third mutation that then enabled it to eat it very efficiently, and that's when it exploded and took off. Lenski went back through the previous generations and found that the other lines would not, even if you ran them many times, wouldn't develop the ability to eat citrate. That if you went back before the 20,000th generation, the bacteria would not re-evolve the ability to eat citrate, at least not with any higher incidence. But after the 20,000th generation, then the rate of re-evolving the ability to eat citrate was much, much higher. So that's how they figured out that there was this sort of pre-adaptive mutation. So this, they're arguing, confirms the notion of historical contingency in evolutionary history.
Right.
This destroys the notion of irreducible complexity. Because you would say, look, you need these three mutations all lining up. One or two of them won't do anything. Any one or any two of them will not be an advantage. It's irreducibly complex. You're right. And the bacteria evolved anyway. This is something we knew, that just from logic, that this is how things can happen. But now it happened in a lab in front of our eyes. It's unequivocal. There it is,
Can you explain to us a little bit more of what genetic drift is?
Genetic drift means it's just random mutations that are not under selective pressures. And the frequency of genes and of mutations in populations can change over time. They sort of drift over time, not being directed in any direction in any way. And this is one means by which populations start to become different from each other genetically, and diversity increases over time. New mutations accumulate, and this shows that kind of random genetic drift can set the stage for further, adaptations.
Maybe you could wrap up this interview with maybe some advantages and disadvantages of using this type of research method?
Yeah, I think the disadvantage of this approach is that it takes decades, right, to get this kind of data. This was a 20-year experiment, and you think about all the different storage and cataloging of these generations of these 12 different lines of E. coli. It's really a Herculean effort. It's 44,000 generations. It's a lot of generations, right? It would be impossible to do the same magnitude of a study with a vertebrate like mice, for example. You could do a much, many fewer generations, but it'd be harder to get this kind of data.
The rest of the New York Times article transitions nicely from the original experiment of producing citrate-eating E. coli into other examples of evolution in the lab. One of note is the creation of cheater E. coli. A colony of E. coli developed a unique method of survival. To crowd out the rest of the E. coli, this colony developed a method of growing into a mat. They produced cellulose, which made them all stick together into a film on top of the flask, drowning out all the other E. coli. After a while, cheater E. coli were developed. The cheater E. coli had the advantage of being part of the colony and being part of the mat, without having the disadvantage of having to produce cellulose. When too many of these cheater E. coli were grown, the entire mat collapsed, making the entire colony lose its evolutionary advantage. This is a great example of how natural selection doesn't always produce the best organism which is best for the species. I found this article very interesting because it went through several different examples of experiments where you can show evolution, natural selection, and genetic drift within a lab setting. This adds another piece to the large amount of evidence that supports evolution. We can see now how evolution works on a scale in the laboratory. Carl Zimmer, the author of this paper, did a very good job in writing this essay. He starts the essay off with a great piece of imagery. We feel as if we walk down the halls into the laboratory at Michigan State University and see the scientist working at his table for 20 plus years with these different lines of E. coli. He paints an image of how Dr. Lenski is a lone scientist kind of on the fringe, trying to prove a certain piece of evolution. Many of his colleagues had very little faith in him that this experiment would work well. He then goes in and shows how great this experiment turned out. The author transitions to several other experiments that resulted from this great line of science. The paper then ends with a great quote where it says, "Mutations like this one," Dr. Velliker said, "make for a much more complicated story. It is a story he and other scientists are looking forward to revealing." This gives a great look into how scientists are always searching for the truth. It's a very nice quote to end the paper with.